All right, it's so great to be here. I'm John Guatelli, and I came from the University of Colorado in 1984, I think it was, to UCSD. And so I stayed here ever since, and Doug is a huge part of the reason why that is the case. And, I guess I met Doug. The first time was when we were on consult service together. Doug, if I remember well, was January of '85, and I was an ID fellow. And frankly, I was just so taken with him. He was so knowledgeable. He had a tactical, logical approach to patients. And, a mentor is someone you want to be like, right? So I wanted to be like that. I'm not sure I made it, but at least I've made an effort. I was very happy to join his lab to do HIV research. And I think we've heard a lot of testimony about what a great mentor Doug is. And one, I think Albert Wu just said, finds things in you, brings out things in you that you might not believe in yourself. And I think that was a great line. I really feel like Doug did that, has done that for me. And I think there's only one person in the audience, that's Chelsea Spina, who's had the great privilege of having an office next to Doug for the last 20 years. And that door has always been open. Well, there's been some conference calls, I guess, where it was closed. And I, as Doug could tell you, wander in at any time of the day, either to tell him something exciting or commiserate about something not exciting. But it's been a really great relationship, and it really has meant a lot to me and to everyone else here. So Doug, I want to mention one thing. There's been a lot of Doug-isms that have come up here today, and one of them, which I don't think you told me when I was a fellow or resident rather, but I've heard it since then, was if you don't know what to do, don't do anything. And I think that was tremendously good clinical advice, and probably advice in general. So Doug suggested that we make this about science, and I hope that I don't overdo it here. But I'm going to talk about two HIV proteins that I've spent most of my career on, which some of you may or may not have heard of, named Vpu and Nef. And the big idea here is that these proteins function by modulating the trafficking, getting other cell proteins off the cell surface, which benefits the virus, and that benefit is in large degree immune evasion, as I'll try to show you. But before I do that, I want to show you the Richmond lab circa 1989. And, close inspection here will show you that members of the audience, like Chelsea Spina and Pascal Melan here, and Doug and I, it's remarkable how nothing has changed in 30 years. And but something has changed in 30 years, and this is also, of course, hugely remarkable. This is just an off-the-cuff list of things I remember as a fellow in clinic in 1989, and these, deadly opportunistic infections and tumors that we struggled with are now gone off the list of things that I see in clinic today. Instead, this looks largely like an internal medicine clinic, with the exception of a few particular syndromes. And of course, we have antiretroviral chemotherapy to thank for that, and we have Doug to thank for that, for all the seminal contributions he made to make this incredible transformation happen. So, as you know, in the absence of therapy, there's continuous viral replication. Cells die producing virus, they infect new cells, and this is a continuous process with a relatively short time of the replication cycle of the virus. But as you've also heard, some viruses go into cells that seem to become quiescent and latently infection, yet they harbor viral genomes that can be produce replication competent virus, and therein is the notion of the latent reservoir. And you've also heard about a strategy for cure, which was dubbed shock and kill, in which these cells that have the HIV genome in them could be induced to produce virus, and then the immune system would help take care of them and eliminate them. And so, here is a made by some immunologists that I borrowed an example of immune surveillance of HIV-1, and I couldn't begin to explain very much of this, but I would like to point out that the idea is to get rid of these cells, to to kill them and get rid of this integrated viral DNA in them. And one way that host immunity does that is with a cell type that are CD8 positive called cytotoxic T lymphocytes. And this is counteracted by one of the genes that I want to describe in detail, Nef, to a lesser extent VPU. And another way to kill cells is with cells that are called natural killer cells. And one of their functions is something called antibody-dependent cytotoxicity, where an antibody recognizes the virus or a viral protein. In this case, it can only be envelope glycoprotein. That's the only thing on the surface of these cells that's virus-specific, and kills them. And this is actually counteracted by Nef and VPU. And the way all these things happen, and I'll just talk about 3 surface molecules that are downregulated by these proteins, removed from the cell surface. And they are CD4, the viral receptor itself, a class 1 MHC that presents viral antigens to the cytotoxic T lymphocytes, and a BST2, and which I'll show you in a second. So here's just the HIV genome, and you know, Paul is a major drug target. You need an envelope, of course, and these structural proteins to make virus, but colored in here are these so-called accessory proteins. And they were named that originally because they seemed dispensable for in vitro replication of the virus. But they all obviously have important functions, or a small genome like this would never keep them evolutionarily. And VPU is specific to HIV-1, whereas Nef is found in both HIV-1 and HIV-2. And here's a schematic of these 2 small viral proteins that are encoded by these genes, VPU and Nef. They're both membrane-associated proteins. And in this diagram, these cylinders are alpha helices. So VPU is a transmembrane protein and has this cytoplasmic domain. And just to remember, if you can, there's a couple of serines of interest here. And then there's this so-called acidic leucine motif. L's for leucine, acidic glutamic acid, the abbreviation is E. And so that's going to be important a little bit later. And Nef here associates with membranes, but in a different way. It's modified at its N-terminus with a lipid myristate group. And then here's the sort of folded structure of Nef, and there's this loop out here that also has a very similar motif, this acidic leucine motif. So how these proteins work, they're not enzymes, they just act as molecular adapters. They will bind their targets, which I'll show you in a second, and they'll also bind components of the cellular machinery that normally traffics membrane proteins. And so just to start with Nef, it removes CD4 and class 1 MHC from the cell surface. And it may seem counterintuitive for the virus to remove its receptor, but remember, we're talking about the cells that are making the virions here. CD4 is actually very bad for the virus in the cell that is making virions. If it interacts with envelope there, many deleterious things happen, one of which I'll try to show you at the end. But anyway, in this experiment, if you look at the relative surface staining with antibodies CD4 and MHC-1, you can see how dramatically Nef will downregulate these proteins from the cell surface. This just shows the same kind of thing in a technique called surface biotinylation in Western blot. So we certainly didn't discover this, but it's a nice experiment that shows that infected cells are sensitized to CTL-killing when Nef is unable to downregulate MHC-1. So what you're looking at here on the right is what happens to HIV-infected primary T cells that have been infected with virus in the lab, and then they're incubated with a cytotoxic T lymphocyte clone. It recognizes this particular epitope shown here in the GAG protein, and this just shows that a peptide of that causes the cells to degranulate, which is how they ultimately kill the cell. But this is the fold reduction in cells that are HIV expressing. So it's kind of backwards. The better the killing is, the bigger the bar. And you can see that if Nef is disabled by a specific mutation, there's much better fold reduction, much better killing of the cells by CTLs. And that's specific if one does the experiment with mutation in this epitope, you don't see much killing. So, there we go. Okay. So both Vpu and Nef interact with a protein family called Clathrin adaptor complexes. So Clathrin is like a scaffolding protein that goes around vesicles that bud from various places in the cell, and these vesicles transport proteins. But between Clathrin and proteins that are going to be transported are these things called adaptor protein complexes, and there's a family of them, but we'll just show a couple today. One is AP2, which is generally involved in endocytosis, taking a membrane off the cell surface, and another is AP1, which is really not fully understood, but probably involved in transport between so-called endosomes and the trans-Golgi network, and also lysosomes. And if we look at what the structure of these adaptor protein complexes, there are these heterotetrameric complexes, and they contain binding sites for the cytoplasmic parts of cellular proteins. This is how cellular proteins are regulated in the cell, and there are different sequences in those cytoplasmic domains that bind different places on these. So there's a binding site for this so-called acidic leucine-based motifs. Remember I showed you Vpu and Nef both have those, and that's basically on this sigma subunit. Then there's sites for something called a tyrosine-based motif. Y is a tyrosine, normally has a bulky hydrophobic residue there, and that's on the mu subunit. And there's also a third type of signal called acidic cluster, which has a run of these negatively charged residues and serines that typically get phosphorylated. So getting back to Nef, Nef forms these ternary interaction complexes that enable the virus to modulate CD4 and class 1 MHC. And if you look at how these have been drawn, they look different because they kind of are different. In the case of CD4, we're coming off the plasma membrane, and Nef seems to interact via that acidic leucine motif with the AP2 complex, remember, involved in endocytosis. Something else to notice are these leucines in the CD4 cytoplasmic tail. They're required for Nef to act on CD4. Whereas MHC-1, I'm showing this interaction in the trans-Golgi membrane and ultimately going to lysosomes. And this is a different kind of interaction. First of all, it's with AP1, different Clathrin adapter, and there's sort of a three-way interface here, and it depends on a key tyrosine in the MHC cytoplasmic domain, which is in this sequence there. So we worked on this for some time and figured out what the right proteins were, and then we had a great structural collaboration that began in 2012, and actually were able to participate in understanding exactly the structure of the ternary complex formed by Nef, the MHC class 1 alpha chain, and this mu subunit of AP1. And the MHC1 cytoplasmic domain is here in purple, and the mu subunit, that's the subunit of AP1, is in green, and here's Nef, its structure shown in all this in a ribbon diagram. And the key thing is, look how Nef basically sandwiches the cytoplasmic domain of MHC class 1 against this mu subunit. And if you look in this model of that, here's the MHC cytoplasmic domain again. You can see the side chain of that critical tyrosine, and it fits in this interesting groove between these two proteins. Something's missing here, though. Remember, I mentioned that cellular proteins have a critical hydrophobic bulky group there, like a leucine, and so that's missing. Like, here's an overlay of a cellular protein, how it would have a leucine that fits in this pocket, and class 1 MHC cytoplasmic domain misses that. It just has an alanine. So Nef makes up for that, first of all, by making this kind of wall with this polyproline region to hold this in place, and then secondly, with other interactions that are electrostatic and involved positive charge residues on the mu subunit. And some of these are kind of straightforward, like this one here, where this acidic cluster, all these glutamic acids in Nef, are near this positive charge region in mu. And this other is more intricate, where there's actually kind of a three-way interaction here between residues in the cytoplasmic tail of MHC1, Nef, and then these are basic residues, positively charged residues in the mu subunit. So an overarching view of that would be something like this. Here would be this complex that's up against the membrane, and MHC1 would not normally bind it because it doesn't have all the right stuff, but Nef brackets it and enables it to bind and then be trafficked in an abnormal way to provide immune evasion. So again, that would be shown here. Normally, viral peptides would get loaded onto class 1 MHC and they'd go to the cell surface. But if HIV expresses Nef, the whole process gets kind of intercepted here, and instead of getting to the cell surface, ultimately they go to the lysosome and get degraded. Something we're working on right now with our collaborators is understanding better the structure of Nef, CD4, and the AP2 complex involved in endocytosing Nef off the cell surface. And this is still not known. How does the CD4 cytoplasmic domain interact here? And this is a tentative model. This shows the entire AP complex, and this little pink part is CD4, and this blue would be Nef. And if you were to rotate it up and look down from the membrane, you'd see it like this. This is the AP complex, and this kind of aqua blue is Nef, and then here's this little bit of CD4. So one of the parts of this that was already known from a smaller structure is how Nef really interacts with AP2 in this way. And this is driven by this acidic leucine sequence I told you about at the beginning. And that really drives this interaction. And then CD4 seems to be completely opposite that. So here Nef acts like a linker to bring CD4 to this AP complex. And we think this is how CD4 fits in this particular pocket of Nef, and you can see these key leucines that I said they were important for CD4 downregulation. This part is still tentative, and it may be that the model isn't quite right. You can see how this mesh is electron density prediction based on crystal structure, and you can see it's not really a perfect fit, but that's our best guess at the moment, and we're trying to validate that by mutations biologically. So the one thing interesting about this model, though, is it would suggest there's a common binding site on Nef for the cytoplasmic domains of MHC-I and CD4. So even though they're very different in how it works, like look, Nef over here recruiting class 1 MHC is over here on this side of the complex, recruiting CD4 on the other side of the complex. But if you look at where Nef would be binding these domains, it's in a relatively common area, though the contact points are definitely different. So I want to switch to VPU and tell you this was a big finding for us in 2008. It had been known for 20 years that if the HIV doesn't express VPU, the virions remain stuck on the cell surface. And so this is a scanning electron micrograph of cell surface of VPU-negative viruses. And you know, I don't show you a control here, but the viruses should get off. They shouldn't be stuck to each other and stuck to the cell surface like this. If you look really closely, you can imagine there might be some connections between the virions. And we were fortunate to identify how this all worked. It turns out that VPU is stimulating the release of virions from infected cells by counteracting a cell surface protein that would otherwise keep them trapped there. And that is named BST2. And it is also an interferon-inducible protein, so it's part of the host response to viruses. And it works on more than HIV-1. It works on a number of envelope viruses that bud from the plasma membrane. So a picture of BST2 would be shown here. It has an unusual protein, two membrane anchors. One is a glycosyl phosphatidylinositol. Usually proteins that have that GPI anchored, they don't have another anchored. The rest of it just floats out in the breeze, so to speak. But this protein has another transmembrane anchor. So it can literally hold the membrane of the cell to the membrane of the virus if it's there at the time of virus budding from the cell surface. Kind of shown schematically again here. And we could demonstrate this with antibodies to BST2 in an electron-dense label that it located to the cell surface between virions and cells and virions and each other. And so, you know, like Nef, VPU decreases expression of cell surface proteins, and it decreases BST2 at the cell surface. So you can see here in this experiment where only VPU is expressed, you look at the expression of BST2, how dramatically down this goes, even if you don't see much change in the level of protein in the cell as a whole. And so VPU is known to also downregulate CD4. There's quite an overlap between VPU and Nef in their targets, and CD4 is one of them. Both of them work on it, and it was known to do so by recruiting a different type of protein complex, something called the ubiquitin ligase complex with multiple subunits based on this protein family called culin. And VPU would bind a cofactor here called beta-TrCP that's part of this complex. And in doing so, it would recruit this protein degradation complex to BST2. So here's a different way of looking at BST2 now with both ends in the cell membrane. This interaction is actually remarkable. It's between the transmembrane domains of the two proteins. So we thought, well, this might be how things work. And so we tried expressing a dominant negative version of this protein. So in other words, something that could only bind VPU but not recruit it to the rest of this complex, which I should have mentioned, the main point of which is to modify the target with ubiquitin, which is a protein that when linked, especially in multimeric form, to a protein will target it to degradation, trafficking either lysosomes or proteasomes. So anyway, we tried to overexpress this dominant negative protein, and it did pretty well at blocking BST2 down regulation if you compare to the shaded to the open here in mock. But interestingly, when we tried a dominant negative version of this part of the complex, it didn't work so well. That's shown down here, and if you just look at I guess the green compared to the red, you can see that for CD4, blocking this part of blocking this activity was quite effective, but not for BST2. It was still down-regulated. And we speculated that Clathrin, these Clathrin adapters, might be part of VPU's activity too. And so that's true. If you look at BST2 down-regulation here, the fold effect, it's much less effective if Clathrin is knocked down, or these, to a lesser extent, these adapter proteins that link targets to Clathrin. And so although AP1, oops, I should show you, doesn't really seem likely to account for enough of this activity, we were able to, with our collaborators, arrive at something of a structure of the AP1 complex along with VPU and BST2. And if you notice, there's only little bits of these proteins that are bound to this complex. And it turns out that BST2 has one of these tyrosine-based motifs. It's a little more legitimate than the one in MHC class 1, right, because it has this hydrophobic residue. But even there, it doesn't really fulfill this binding pocket so well. Whereas VPU has one of these acidic leucine motifs, very similar to Nef, and I showed you how Nef bound to AP2 via that type of motif. So the idea then is that VPU would bind BST2 via the transmembrane domains here, and that you have you have a cytoplasmic sequence in VPU binding one part of this complex and BST2 binding the other, and that would lead to the trafficking of BST2 to the wrong location, off the plasma membrane. And this is very different, in comparison to how Nef brings MHC-1 into the very same complex. So I want to end just by trying to show you how some of these things might have consequences for cell killing by ADCC, which I mentioned at the beginning, antibody-dependent cytotoxicity. So if you imagine an infected cell surface here, and the virus is expressing VPU and Nef, the virions get away. We don't even show one here. They're gone. But there is going to be some degree of envelope on the cell surface, and that can be recognized by antibodies. But envelope-trimers, you may know, are typically in a closed conformation when they're on cells and initially when they're in virions. That conformation opens up when they bind CD4 and they begin to undergo changes that allow the virion to enter a target cell. But also on this infected cell surface, in the presence of VPU and Nef, you'll have less BST2 and CD4. But if you lack these proteins, VPU and Nef, a couple things are going to happen. For one thing, these virions get trapped, like I explained. And so all this envelope protein that's there will be a better target for antibodies. Moreover, because there's no CD4 downregulation by any of these, either of these gene products of the virus, envelope has a chance to interact here with CD4, and that causes opening of the trimer and accessibility to antibodies that normally wouldn't recognize it, and that should in principle enhance killing by ADCC. And the last data slide I'll show you shows that this is true, that VPU and Nef in effect decrease cell surface envelope epitope exposure, and thus they inhibit ADCC. And here cells are staged for CD4 BST2 and an antibody that's specific for CD4 induced epitopes, or with HIV IgG pooled from patient plasmas. And if you look at cells that lack infected with cells that lack Vpu and Nef. There's more CD4, there's more BST2, there's much more staining for this epitope that's CD4 induced, and there's much more staining, ability to recognize cells by patient plasma. And similarly here, if you look at an ADCC assay, this is a concentration, increasing amount of pooled plasma from patients, and this is just the fractional survival of cells. Here with increasing antibody, the cells are being killed, but they're much better killed if they lack Vpu and Nef. So I hope I've been able to show you that HIV, Nef, and Vpu are viral countermeasures to aspect of host immunity, and that these proteins function partly by co-opting Clathrin adapters and related endosomal sorting machinery to modulate cell surface proteins, among them the one I emphasized. Despite their structural similarity, Nef and Vpu share binding interfaces on these cofactors, the Clathrin adapters, and this convergence in how they work probably relates to mimicry of cellular proteins. And getting back to the notion of cure and antiviral chemotherapy, I still hope that ultimately we could find a way to inhibit these proteins, because if we could, we might be able to facilitate the clearance of infected cells and contribute to cure. So I just want to acknowledge everybody, many of whom data I showed you, and current members of the lab whose data I wish I could have shown you are doing very exciting things. And it's really a great pleasure to be here and celebrate Doug. So thank you.